Nucleus. Certain eukaryotic cells such as the mature sieve tubes of higher plants and mammalian erythrocytes contain no nucleus. It is the largest cellular organelle in eukaryotes. Prokaryotic cells lack nucleus and is complemented by nucleoid. In mammalian cells, the average diameter of the nucleus is approximately 6 micrometer, occupying about 10% of the total cell volume. The contents of the nucleus are DNA genome, RNA synthetic apparatus, and a fibrous matrix. It is surrounded by two membranes, each one a phospholipid bilayer containing many different types of proteins. The inner nuclear membrane defines the nucleus itself. In most cells, the outer nuclear membrane is continuous with the rough endoplasmic reticulum, and the space between the inner and outer nuclear membranes is continuous with the lumen of the rough endoplasmic reticulum. The two nuclear membranes appear to fuse at nuclear pores, the ring-like complexes composed of specific membrane proteins through which material moves between the nucleus and the cytosol. It contains cell's genetic material, organized as multiple long linear DNA molecules in complex with histones, to form chromosomes. The genes within these chromosomes are the cell's nuclear genome. The function is to maintain the integrity of the genes that controls the activities of the cell by regulating gene expression. In a growing or differentiating cell, the nucleus is metabolically active, replicating DNA and synthesizing RNA, tRNA, and mRNA. Within the nucleus, mRNA binds to specific proteins, forming ribonucleoprotein particles. Most of the cell's ribosomal RNA is synthesized in the nucleolus, a subcompartment of the nucleus that is not bounded by a phospholipid membrane. Some ribosomal proteins are added to ribosomal RNAs within the nucleolus as well. The finished or partly finished ribosomal subunits, as well as tRNAs and mRNA-containing particles, pass through a nuclear pore into the cytosol for use in protein synthesis. In a nucleus that is not dividing, the chromosomes are dispersed and not dense enough to be observed in the light microscope. Only during cell division are individual chromosomes visible by light microscopy. In the electron microscope, the non-nuclear regions of the nucleus, called the nucleoplasm, can be seen to have dark and light staining areas. The dark areas, which are often closely associated with the nuclear membrane, contain condensed concentrated DNA, called heterochromatin. Fibrous proteins called lamins form a two-dimensional network along the inner surface of the inner membrane, giving it shape and apparently binding DNA to it. The breakdown of this network occurs early in cell division. Functions of the nucleus. Speaking about the functions of a cell nucleus, it controls the hereditary characteristics of an organism. This organelle is also responsible for the protein synthesis, cell division, growth, and differentiation. Some important functions carried out by a cell nucleus are: 1. Storage of hereditary material, the genes in the form of long and thin DNA, deoxyribonucleic acid, strands, referred to as chromatins. 2. Storage of proteins and RNA, ribonucleic acid, in the nucleolus. 3. Nucleus is a site for transcription in which messenger RNA, mRNA, are produced for the protein synthesis. 4. Exchange of hereditary molecules, DNA and RNA, between the nucleus and rest of the cell. 5. During the cell division, chromatins are arranged into chromosomes in the nucleus. 6. Production of ribosomes, protein factories, in the nucleolus. 7. Selective transportation of regulatory factors and energy molecules through nuclear pores. As the nucleus regulates the integrity of genes and gene expression, it is also referred to as the control center of a cell. Overall, the cell nucleus stores all the chromosomal DNA of an organism.